Before we begin, we would like to acknowledge the Dharawal, Yuin, and Wadi peoples as the traditional custodians of country in the Illawarra. We pay our respects to elders past, present, and emerging, to all Indigenous peoples living in the region. And we extend that respect to the traditional custodians of the lands from which you are joining us today, as well as to other Indigenous peoples with whom we engage. Through our research and engagement, we seek to help others appreciate and act in respect of the significance of country. Good morning, everyone. My name is MK. I am a student of the Centre here at the Wollongong Node, and I'll be your chair for today's seminar. Now, before we start, just some housekeeping announcements. Our speaker, Jianping, will have 45 minutes to present, which will then be followed by 10 to 15 minutes of questions from the audience. Now, we do ask that you save your questions until the end of the presentation, but feel free to type them in the Zoom chat and we'll address them during the Q&A session. Also, a reminder that this seminar is recorded, so please make sure your mics are muted throughout the presentation. And if you're interested, this recording will be available on the CCMMP website afterwards. Now, joining us for this session is Assistant Professor Jianping Wu from the School of Life Sciences at Westlake University. Now, Dr. Wu completed his Bachelor of Science and PhD degrees from Tsinghua University in 2012 and 2017, respectively, after which he then joined Princeton University to work as a postdoc. He has spent more than 10 years studying membrane proteins and was the first to report the highest resolution cryo-EM structure of the eukaryotic voltage-gated calcium ion channel, CAV11. He now works at Westlake University as assistant professor, where his lab works on understanding the protein-protein complexes that are essential during the fertilization process. We look forward to hearing more about it today as Dr. Wu presents on the structural elucidation of CatSper by cryo-EM. The floor is all yours, Dr. Wu.
Okay, so thank you very much for your kind introduction. So first, I will also thank for the organizer for inviting me to join this very wonderful seminar session, and especially thank to Yan Jiang, who is currently a postdoc fellow in Monash University, and who make this connection between me and this seminar. So today, I'll be very happy to share our recent work on the structural elucidation of the CatSper by cryo-EM. So I would like to start with this fascinating picture. So illustrating the very first moment of our life. So this is the fertilization process, which is the event of the merge between a sperm and egg. So this is a really fascinating biological event that marks the beginning of life. So when I set up my own lab, I start to trying to start this elucidate working mechanism between all those processes using structural biology studies. So here shows a general fertilization process of it. Basically, it includes three parts. First is the before the sperm meet with the egg, the sperm need to be activated. So that's the sperm activation process, or also called sperm capacitation. And after that, when the sperm meet to the egg, they need to recognize it with each other. So this is a really important event, especially for the organism in the water environment, that the sperm and egg are in the open environment. So that they must recognize with each other of the same species. And after the merge and the recognition of between the sperm and egg, the sperm will release the enzyme that can digest digest the very thick layer of protection layer of the egg called zona pellucida, and get into the perivitalized space. And after that, the sperm membrane and will then fuse with the egg membrane and release the nucleus into the egg for successful fertilization. So the sperm, it has a very unique structure. So basically, it contains three main parts. The head, which is contain a very condensed nuclear with DNA, that is the genetic information, and the middle piece, which mainly contain the mitochondria that can provide energy for the movement of the sperm, and a very, very long tail, which has contain the axonal microtube based axonal that can provide the motion of the sperm. So the sperm can, although the size of sperm is very small, but it can be as long as 40 micrometer long, and its swimming speed can be as fast as 0.75 meters per hour. So this is almost over 5 times of the length of its body length. So think about it, it is actually much faster than the swimming speed of human. Because this is because it has very unique structural features. So the sperm activation process is highly controlled by calcium signaling, and so basically the mature sperm only has very basal motility that cannot fulfill successful fertilization. So in order to fulfill the successful fertilization, the sperm must be activated. That's called sperm capacitation. And one major hallmarks of capacitation is the hyperactivation, which the sperm flagella tail can undergo very robust beating and asymmetrically movement of the tail. And this process is highly controlled by calcium signaling. And one major ion channel located on the sperm membrane is called the cation channel of sperm, which is short for CatSper. And this protein is the sole cation ion channel that is responsible for the diffuse of the calcium ion into the sperm tail that can trigger the downstream signaling that in turn triggers the hyperactivation of the sperm. So this ion channel is important for the calcium signaling of the sperm, and with this mechanism, it can help to the sperm to respond to the environmental stimuli. For example, the hormones, the progesterone, which is the major hormone that can activate the sperm. So this is because this hormone can bind to lipid hydrolysis, and this can activate this lipid hydrolysis to hydrolyze a lipid, which can ultimately activate CatSper to diffuse the calcium ion into the sperm tails. And also pH, temperature, and memory potential, all these environmental stimuli can work on CatSper channel to activate this. And the CatSper is the first identified more than 2 decades ago. So the first component of CatSper is CatSper1, so which is identified in 2001 by David Clapham lab. And so we can see that after knockout of CatSper1, so the sperm motility is affected. So normal sperm can undergo hyperactivation with this unique asymmetric robust beating of the sperm flagella tail. But when knockout of the CatSper, it cannot undergo hyperactivation. And this CatSper knockout can also affect the fertilization process, which So here shows the Y-type sperm. It can have this successful fertilization. So after fertilization, the fertilized egg can undergo cell division, but mutant sperm, the egg cannot be fertilized. But when we remove this zona pellucida protection outside of the egg, even the mutant sperm can undergo successful fertilization process. So this highly indicates that CatSper is really essential for the very beginning of the sperm activation process, but not the fertilized the final fertilized process. Okay, so after the first after the identification of the first component, there are more than 10 components identified that have been reported to be the component of this super complex. So CatSper is currently the most complicated ion channel so far, and so among those components, CatSper 1 to CatSper 4 is the iron conducting subunit, which can constitute the iron conducting pore. And all the other components are, we can see that they are auxiliary subunits that combined to the peripheral of the channel, which may help to facilitate the membrane location and provide regulating roles for the channel activity. And despite of that, in the past few in past 2 decades, people failed to reconstitute this protein and record the current of CatSper in heterous expression system. So that also indicated there might be remaining unidentified component of this huge complex, and this remaining unidentified components of CatSper might also be important and essential for the activity of functional CatSper. Okay, so CatSper 1 to 4 is actually belong to a large superfamily called the voltage-gated iron superfamily. So this is a large superfamily of a membrane protein that also include many other ion channels. For example, the voltage-gated potassium channels, the chip channels, and the two-pore channels, and also the voltage-gated sodium channels and the calcium channels. And all those channels, they share a very common structure fold or functional fold. So which is a module contains 6 transmembrane helices. We named it S1 to S6. So S1 to S4 constitute the voltage sensing domain, and that's why this superfamily is called a voltage gated ion channel superfamily. And this is the structural basis for the voltage sensing, and for channels like sodium channels, calcium channel, potassium channels. So in their S4 helices, there contain several positively charged residues that are essential for the voltage gating of those channels. And S4, S5, and S6 of the channel constitute the iron conducting pore. And there is one interesting point is that for potassium channels, it is usually constituted by it is usually formed as a homotetramer, that which means it is formed by 4 same proteins with those 6 transmembrane helix module. But for voltage-gated sodium channels and the calcium channels, it is actually one single polypeptide chain, which contains 4 homologs but not identical functional repeats. So it also contains 4 so we call it the 4 multiple 6 TM family. And there's also a channel called 2-pole channel, which is might be an evolutionary intermediate of the homotetramer and monomer channel. So those channels, they contains two functional repeats, and so they forms a homodimer. So there's one interesting. Sorry. So here I shown several structure of this superfamily, and here shows the homotetramer structure of a potassium channel, and this is a structure of voltage-gated sodium channel. And this is a voltage-gated calcium channel, and the voltage-gated sodium channel and calcium channel, except for those single polypeptide chain ion conducting subunit, they always also contain several auxiliary subunits, like the beta-1 subunit in sodium channel, and alpha-2 delta subunit, and the beta-gamma subunit in the calcium channel. And there is one interesting question about the organization of those channels, is that CatSper is actually formed by four different polypeptide chains. So here comes to an interesting question about how those 4 components of CatSper are organized in space. So for potassium channels, we have also mentioned, so they form this homotetramer, so there's no such problems. And for 2-pole channels, so they form homodimers, and so they all adopt those domain swap configuration. So that here shows the voltage sensing domain, and this is the pole domain. So the same color represent the same functional repeat. So there is those domain swapped configurations in the pole region and in the voltage sensing domain region. But overall, so they are within the same polypeptide chain. The 2 functional repeats are close with each other. And here is the single polypeptide chain of the sodium channel and calcium channel. So they organize like a clockwise organization from the extracellular view. But for CatSper, because it is formed by 4 polypeptide chain, so they can be organized in like a clockwise organization. So they can also be organized like this one, 1, 2, 3, 4, because there is no limitation or restriction because they are not connected, so they have no this restriction in the organization. So basically, there are 6 possibilities for such organizations of the 4 components between among CatSper 1 to CatSper 4. So which one is correct, or how those 4 components are organized in space is a really interesting questions, and which may be answered only by structural study. Okay, so as I've mentioned, so in the past few years, the current mediated by CatSper has not been a successful record in heterosexual system. So in the past few years, people know the functional or electrophysiological property of CatSper are mainly achieved by the patch clamp recording from intact sperm. So this is a technique developed in 2006, and so by measuring by patch clamp to the intact sperm, so people successfully measuring the current mediated by CatSper ion channel. And here shows the CatSper the current of the CatSper and from a white type sperm, and in CatSper 1 knockout sperm, so this current is disappeared. So that suggests that this current is indeed mediated by CatSper ion channel. And so based on this system, we can study the electrophysiological properties of the CatSper. So basically, CatSper have a low voltage sensitivity. So compared to voltage-gated sodium channel and the calcium channel, so their activation voltage is higher. And also there's a unique property of CatSper is that it can be activated by higher pH. So as this figure shows, when the pH is increased, it shows a left shift over the JV curve. And another unique features of CatSper is the localization in the spur tail. So here shows a super resolution study of the localization of the CatSper. As we can see that CatSper is not random distributed on the sperm tail, but they form like this quadrilateral calcium nanodomains. And later on, people achieve even higher resolution of these images. And we can see that actually in each row of this quadrilateral calcium domains, there are actually have two small rows of this localization. So that indicate that there are 8 small rows of CatSper located on the sperm tail. And this is a really interesting features, and why CatSper has and how they can form such unique spatial organizations in sperm tail is largely unknown so far. So although, as I have shown, although CatSper is essential for sperm motility and fertility, there are still many fundamental questions remain unanswered. So for example, how many components of CatSper are there? There still is still open questions. Although there are already 10 components have been identified, it is still indicating that there might be other new components. And also, how do CatSper 1 to 4 form a tetrameric channel that can mediating calcium ions, and how the whole CatSper complex is assembly, and also what's the gating and the regulation mechanism of this unique channel. So basically, after I set up my own lab in 2018, I started working on the structure elucidation of CatSper and trying to answer all those questions by structure study. So basically, we start from a recombinant expression of this huge complex as usual, and although we know that it is challenging, we start to optimize the expression system, and here I shown a successful case that actually we can recombinant express the key component of CatSper, and we add a fluorescence tag onto CatSper 1 to 4 and the auxiliary subunit beta, gamma, delta, and epsilon, and we can successfully detect all those components. And one promising feature is that those components are actually co-migrate with each other. Although the gel filtration profile does not show very perfect protein behavior, but those co-migration give out some confidence that those proteins might be co-folded with each other and might be suitable for following structural studies. So we tested those samples by negative staining, and this image shows a representative image of a negative staining, and we do see some protein particles, but unfortunately the overall quality is not very high, and as we can see that the particles are not very homogeneous. And despite of that, we collect some data and cryo-EM data and make a 2D classification average analysis. And as we can see that we can see some particles that contains a very representative detergent micelle disk, and that indicates that there is indeed some particles that may represent the CatSper channels. However, the overall quality is just not very good, which cannot help us to achieve very high resolution reconstruction of the channel. And also, as we can see, the best class, one of the best class is actually a proteus particles, which is a contaminant during protein purification. But those also indicate that the protein might not be folded very well and tend to be degraded by the proteus. So this indicates that the recombinant expressed proteins of CatSper may not be suitable for structural study. So now, so after that, we think about maybe this protein is too complicated, and also there might be other essential components for proper assembly of this huge complex. And so maybe purification from endogenous tissues would be a better way to get a well-folded good behavior protein sample. So we tend to, based on this idea, so we tend to the endogenous purification of CatSper. And so we look into the literature and find that the expression level of CatSper on sperm is actually not very high. So basically there are only about 1,000 to 2,000 copies of CatSper on each sperm. And so this is a relatively low expression level of CatSper on the sperm tail. And so in order to achieve native purification, endogenous purification of a native CatSper, so we must think about a way to efficiently purify the protein from this tissue. So we think about an idea to adding a tag onto one of the components of the complex. So that's why we designed a transgenic knock-in mice. In this mice, we knock in 2 tags, affinity tags, or the 3 plus flag, 3 multiple flag and GFP tag before the N-terminal end of the CatSper1 subunit. And so we can verify those modification by genotyping, and we can also observe the sperm of the knocking from the knocking mice. And as we can see that we can clearly see the GFP fluorescence signal in the principal piece, which is right after the middle piece of the flagellar tail of the sperm. So this indicates that the knock-in is successful, but this is not only indicated the knock-in is successful, but also illustrate that this modification does not affect the sperm motility of the sperm. And also we also test the fertility of the knock-in mice, and the result shows that this knock-in mice has the same fertility rate compared to the wild type. So this indicates that this modification of adding a tag on at the N-terminal of CatSper1 does not affect the function of the CatSper channel. So with this, we start to purify the protein by using knock-in mice. So we use 2 two-step of affinity purifications. So first, we utilize this GFP tag. So we use GST fuse the GFP nanobody to bind with this GFP tag. So that after those affinity purification by the GS4B resin, the elute and was then applied to a second affinity purification to go through anti-flag beads. So after those two affinity purifications, we successfully get a purified native sample of CatSper of CatSper complex, and as we can expect it, because those is from the tissue sample, the overall quantity of the sample is very low. So as we can show here, the peak is extremely low, and we so here is a cross-linked sample of the CatSper complex, and we can only visualize the we can only clearly visualize the band on the SDS page by silver staining. However, despite of this low quantity, it is still enough for making a cryo-EM grade. So this is a powerful this is one of the powerful point of the cryo-EM, because compared to X-ray crystallography for structure study, cryo-EM only need a small quantity of a protein sample, and here shows a negative staining of the CatSper sample, and we can see that compared to previous recombinant recombinant purified sample, the overall quality of this sample is more homogeneous. And with this sample, we send it for mass spec analysis, and we can detect all the previously reported 10 components of CatSper, which is shaded in yellow here. And we can also detect many other proteins that may be co-purified or as a contaminant proteins. And this mass spec list was also helped us as a searching pool for the identification of the new component, as I will mention later. And with this, we make a cryo sample for high resolution structure elucidation. So here shows the cryo-EM image, and as you might see, can clearly see here is a protein particles which is inside the view. Here is the transmemory regions, which the features with the detergent micelle feature. And after data processing, here shows the 2D class average. We can clearly see the features, the secondary structural features through the 2D class average. And if we compare those 2D class with the previous result from the recombinant expressed proteins, we can see that they are actually very similar, but the protein from the native source are larger in the transmemory regions compared to here. So here it seems that there's a broken region in the transmemory region, and this turns out to be that there are several new components in the transmemory regions, and which might be essential for the overall assembly of the CatSper. And this immediately explain why previous recombinant expression proteins cannot successfully get a very high resolution reconstructions, although the overall shape is quite similar. Okay, so after data collection, we perform a cryo-EM analysis, data processing, and the data processing is relatively routine. So because CatSper has very large soluble region with very large extracellular domains contributed by the auxiliary subunit CatSper beta to epsilon. So this can greatly help us to align the particles, and to so So after 3D classification, we pick out about 560,000 particles, and this already help us to get a overall map at 2.9 angstrom resolution. However, because this protein part, this complex is so large, at some peripheral regions, the overall resolution is still very low, although the local resolution is still very low, although the overall resolution is high enough. So in order to improve those peripheral regions, so we apply local classifications and the refinement. So the basic idea is that we apply a few local masks, and we extract we extract the particles of those local regions and apply local classifications. And through these strategies, we can finally improve the local resolutions of those specific regions, and finally get a higher composite map for model building. Yeah, so this is the overall reconstruction map, which is contributed by the overall map and also several local maps. And here, each color it represents one component. So we can clearly identify all the previous reported 10 components: CatSper 1 to 4, beta, gamma, delta, epsilon, and also two soluble cytosolic proteins, CatSper eta and EFCB9. And besides that, we identify a few new components in the transmembrane regions, as we can already see from the 2D class averages. So so one is SLC06C1, and the other two are located on the opposite side. So the overall resolution of CatSper of this reconstruction enable us to assign and build atomic model for the majority part of this complex, except for a few regions in the cytosolic side. So here I showing a movie of the whole complex. So here is the transmembrane region, which contains the CatSper 1 to 4, as we can already clearly see that from this map. So this is the voltage sensing domain, and the four components of CatSper 1 to 4 form a pore regions at the very center of the complex. And the unique features is that the larger extracellular domains of CatSper beta to epsilon forms a very rigid complex outside CatSper 1 to 4, and this overall architecture is unique and has never been observed in any other voltage gated ion channel before. Okay, so since we identified several new components, so this is really a surprising discovery of our structure, especially one component, SLC06C1. So this is actually a transporter proteins, and we never thought that there is a transporter and a channel can be stay together to form a huge complex. So after the identification, we perform co-localization, immunofluorescence co-localization to verify those new components. So here shows the previous identified, some of the previous identified components. So they all localized on the principal piece of the sperm tail, and here shows the three new components that were firstly identified in our structure, and they also show the same patterns of localization in the sperm tail. So this is also so this helped to verify those component localization, and how those exciting structure can provide us answers to the previous mentioned questions. So actually, after the elucidation or after the determination of the structure, so it immediately answered the one of the questions is that how CatSper 1 to 4 are organized. So basically, because the pore region, which is at the very center of the structure, the overall resolution is as high, is beyond the 3 angstrom. So which allow us for de novo building of those 4 components. So we can unambiguously build the side chains of all those 4 components, and so after assignment of those 4 components, we can see that those 4 components, CatSper 1, 2, 3, 4, 5, 1, 2, 3, 4, they are organized in a counterclockwise arrangement in the extracellular view, and they also adopt this domain swap structure features, which is quite similar to previous voltage other voltage-gated ion channels. So we also analysis the ion conducting paths of the channel formed by CatSper 1 to 4, and we can see that this is actually the channel is actually in a closed configuration. And the detailed analysis of channel has indicated several interesting points, which might provide insight for the gating mechanism of this channel. So we observe a pair helix in each of the S6 helices, and this pi helix on the S6 helix have been observed in some other channels, for example, the voltage-gated calcium channels, and also some chip channels. And in previous study, people have observed the conformational change of those pair helix to alpha helixes, and which might be a trigger, which might be help to trigger the conformational change of the channel state. And so this, although our structure, the CatSper channel we captured is mostly in a closed state and with a single conformation, and this observation already highly indicate that the channel opening might be coupled with those conformational change of the pi helix into alpha helixes, and also the selective. So within between the S5 and S6 helixes, there's a 2 pore helixes that support for highly conserved negatively charged residues, which is called the selectivity filter. And so this 4 residues is quite important and essential for the calcium recognition and diffusion. And the overall structure features of CatSper 1 to 4 is quite similar to the voltage-gated calcium channel, and this helped to explain why CatSper has a ability to transport calcium ion. And we also observe in the selective filter, we also observe two severe densities that might be represent the bonding ions. So we performed MD simulation of this selective filter regions, and we can clearly see that there are two stable ion binding sites in the selective filter, no matter in the environment no matter sodium channel and calcium channel we add in the environment in the simulation parameters, and so this suggests that, and those two iron binding sites are quite consistent with the structure of observations, and this suggests that there are two conserved iron binding sites that are essential for calcium binding and recognition that help those channel to diffuse calcium ion.
Hi, Jianping.
Hey.
I'm very sorry to cut in. You're nearing the end of your 45 minutes.
Oh, sorry. Okay, sure. Yeah, I will hurry up. Okay, so sorry, just skip the voltage sensing domain part. So there's interesting point is that there are four component CatSper beta, gamma, delta, epsilon, which form a very large dome above CatSper 1, 2, 3, and one interesting point is that the interaction between gamma and the epsilon to CatSper 1 and 3, the binding mode is quite similar to the toxin binding to the sodium channel. So this immediately indicates that those components might be helped to protect CatSper 1 to 4 to avoid the attack by extracellular by environmental toxins. And if we observe those assembly patterns between of those large auxiliary subunits, that we can see close interactions of CatSper gamma and epsilon, but there are relatively far away distance between beta and epsilon to CatSper 1 to 4. And the reason for those large distance is because there are several new components that sit aside those two auxiliary subunits. And this is a really exciting observations. And as I've just mentioned, one of those components is a transporter, and this is perhaps the first channel transporter supercomplex that was structurally observed. So that's we named a new name for those supercomplex called CatSpermasome. And on the other side, we identified actually there are three new components, and we successfully identified the two of them. One is CatSper eta, and the other is 249 TMEM 249. So one interesting point is that recently there is a cryo-ET study of CatSper showing spatial organization of the CatSper on the sperm tail is like a zigzag mode, which means that CatSper can actually interact with each other to form a higher order of organization in the sperm tail, and it from this low resolution image we can still see that the interaction point between those CatSper two CatSper channel is actually where the new identified component located. So we think that those new components, it might be essential for those organizations of the higher order of the CatSper. Okay, so there are also a very interesting observation in the cytological regions that we can see there are very long S6 helices of CatSper 2 and 3, which is sandwiched by unidentified densities, and we think that those cytological regions, which is responsible for calcium binding, might be triggers potential conformational changes of the S6 helix, and ultimately induce the conformational change of S6 6 helix to induce the open of the channel. So so we think that the elongated S6 helix can evolve apply to alpha helix transition to undergo a potential activation process of the channel. Okay, so this is the last slide for a very brief summary. So we report the first high resolution structure of the native CatSper, and we surprisingly find that this is actually a transport channel ultra-complex, and that we named CatSpermasome. And the structure provides a basis for the overall assembly, ion selectivity, voltage sensing, and the potential gating mechanism of this supercomplex. And last but not least is that it opens many new opportunities for future studies. For example, what's the function of those new components to the CatSper channel, and also it provides a layer foundation for future drug development targeting to CatSper for male infertility treatment. And our study has been highlighted by a news and reviews written by David Clapham, who first identified the first component of CatSper. So I think this is a really exciting moment for me. And with that, I would like to thank the team. So this work was mainly by this team. So here, this is Meng Ke, Shi Yi, and Professor Zhen Yan, and this is me and Yu Qi. So I'm so proud that we are a very young team, and with average age below 30, and so this is really exciting for us to make a very huge achievement for us. And this is our whole team. And this work was mostly funded by Westlake University and Westlake Laboratory. And with that, I would like to thank you for your listening. Okay, so thank you, and I would like to take questions.
Thank you so much, Jianping, for an amazing talk, and congrats for elucidating such a beautiful and unique ion channel structure. We'll now open the seminar to questions from the audience. So if you have questions, raise your hand, or you can type them away in the chat. Yes, Gokhan.
Hello, can you hear me?
Yes.
Great. Very nice talk and on a very challenging sample. Well done. So what I was wondering is, what was the smallest area when you were doing focus refinements? What was the smallest size of the smallest area that you increased the resolution for in kilodaltons, more or less?
In kilodalton. So let me go back to. So this. Right. So I guess
whichever is the smallest region, yeah.
I guess this one, maybe this one is the smallest region. So only contain a few transmemory helices. So it's not very big. Maybe like 50 to 100 kilodalton.
Okay.
Yeah.
All right. Thanks.
Yeah, but one point is that for some cases, so in order to achieve a very efficient classification, so probably especially for small regions, so we try to apply classifications without alignment. So that's mostly used in rely on so with the option like that is skip align. So we can apply the angle already get from the refinement procedure. And so with this option, so sometimes the classification is efficient, which can help us to classify the very best class to improve the local resolution.
All right. Yeah. Thank you.
Yeah. Thank you.
Yan Jiang, you can ask a question.
Hi, Jianping. Very nice talk. I have a question. Like I saw you use a very small amount of this protein pull out from endogenous, and how much is the concentration final like you apply to do the cryo-EM? Do you use like a special like EM grades for your study?
Okay, so thank you for your question. So this is a very good question. So that many people ask me, yeah, because this is the overall sample quantity is so low, and that we are almost do not have this do not have extra protein sample for measuring their concentration. So that means we don't actually to measure the concentration, but we can roughly estimate the concentration. I guess is like a few maybe 50 to 100 nanogram in total of the protein sample, so because of the low overall quantity and concentration, so Ian asked whether we make a effort in the cryo sample preparation. Yes, so in order to improve the cryo sample concentration, so we applied we use the grid with thin carbon film coated, and that can help us to increase the sample concentration, the particle concentration on the cryo-EM grid. Yeah. So despite of that, as you can see here, the particle density is not very high. It's still not very high. Yeah.
Thank you, Jianping.
Jack?
Hi there. Can you hear me okay?
Yes.
Yep, awesome. Thanks for a really great talk. That was fantastic. I actually just had a quick question about that extra transporter protein you found, SLCO61.
Yeah, okay.
Yeah, can I ask, presumably when you found that protein, were there any other sort of analogous or like closely related proteins you found in the human genome that might also be present in your supercomplex?
Mm-hmm. Wait, sorry. So sorry, you asked too fast.
Oh, I'm sorry.
You repeat your question?
Yes, of course. When you found the SLCO61 protein, did you have a look, I suppose, at the human genome for other closely related proteins? Are there any with enough sequence similarity you think they might be able to interact with this CatSpermasome, with this really big complex and that higher order chaining event happening?
So you mean, is there any other close related transporter that might be similar to those protein?
Yeah, exactly.
I see. Okay, so this is a very good question. So actually we take some time to identify this component. As you can see, the overall resolution, the local resolution is not extremely high for side chain assignment. So basically, we have several criteria for those assignments. So we do search for the proteome database of the whole sperm. So there are actually not many SLC channels, and we predict all the structure of SLC transporter to see whether their predicted structure is similar to the overall density, and this is one key criteria. And for most SLC proteins, they don't have this very unique CASA domain. So they only have those two domains, N domains and C domains. And those extracellular CASA domain is a key unique feature of the identical component, because we can clearly see an extra density here, which can be perfectly fit to the CASA domain. So this is one point, and the other point is that we, as I mentioned, we identify we analyze the mass spec result of the purified sample, and SLC601 is ranked really so I guess it's ranked it's ranked very at the very top of the list. And so this also indicates that this might be the right assignment. And one more clue is that there are previous functional studies that using knockout CatSper to compare the protein profile with the white type, and they can see that the CatSper related protein are diminished or the expression level is lower compared to the white type, and those also include SLC6C1. So all those evidence together strongly support those assignments.
Right, thank you.
Yeah, okay.
Well, I don't see any more questions. So with that, I think we've reached the end of the seminar. Thank you so much, Jianping, for joining us this morning and for your amazing talk. And thank you also to everyone for joining today's seminar. We'll see you all at the next one. Have a great day.
Okay, thank you. Have a good day.